Hi, my name is Mykhaylo Slobodyanyuk, and today I'm excited to present my poster titled Direction-Aware Data Fusion Techniques for Multi-Omics Pathway Enrichment Analysis and Biomarker Discovery. So to begin with the background, different omics techniques allow us to characterize the layers of the cell, such as the genome, the epigenome, the transcriptome, proteome, amongst others. And all these layers of the cell provide distinct yet complementary information about cell function. They don't function individually or independently, but they communicate with one another and influence each other in various ways. That's why it's crucial to jointly analyze these datasets to obtain a holistic understanding of cell biology. Previously in the lab, we've developed an R package called Active Pathways, which is an integrative pathway enrichment analysis method that uses data fusion to merge signals from multiple omics datasets. First, various upstream analyses such as differential gene expression are performed and p-values are acquired. These significance estimates are well suited for data integration because the upstream processing is expected to take care of technical and experimental confounding effects. As input to active pathways, we provide a matrix of gene p-values across multiple omics datasets and a GMT file of functional gene sets such as gene ontology biological process terms. A p-value merging method, such as Brown's or Fisher's, is then utilized to fuse the various datasets into a single merged gene list. A ranked hypergeometric test is then performed between the functional gene sets and the merged gene list to identify enriched pathways. And lastly, the contribution of each individual input dataset is then evaluated to see whether the pathways that were identified from the merged gene list could have been also identified from each individual dataset alone. Currently, Active Pathways accepts only p-values as input to perform data integration. This is problematic as different datasets may also contain directional information, for example, fold change information from differential expression. Inspired by the METAL method, which is an extension of Stuffer's method that has a directionality term, we've also extended Fisher's method to incorporate direction. So to summarize, in panel B, we've incorporated this directional logic into Active Pathways, where in this example, we're integrating RNA and protein datasets by prioritizing genes where mRNA is consistent with protein, such as gene 1 and gene 3, and penalizing genes where mRNA is inconsistent with protein, such as gene 2 and gene 4. By doing so, we will be removing false positive signals and improving gene and pathway level detection. As our first case study, we integrated HOXA10AS transcriptomic datasets. So previously in the lab, Isayev and others studied this long non-coding RNA called HOXA10AS, and they performed knockdown and overexpression of this gene in a glioma cell line. Wild-type, knockdown, and overexpressed transcriptomes were profiled, obtaining p-values and fold change data from the differential expression of wild-type and knockdown and wild-type and overexpressed conditions. We expect genes that are upregulated upon HOXA10AS knockdown to be downregulated upon HOXA10AS overexpression. If this is not the case, we penalize those genes that have the same directionality, as these likely arise due to noise or are not influenced by HOXA10AS. If we look at the plot on the left, in panel A, we have the merged p-value when direction is ignored called original on the x-axis, and the modified method is when direction is accounted for. This is an merged p-value. Dots are individual genes from this analysis, and if they lie directly on the line, they are not penalized. But genes that fall under the line are penalized when direction is accounted for. We can see that out of the significant genes, the ones that are beyond the vertical dashed line, 115 lose significance when penalized for directional inconsistency, and 1,024 are prioritized. If you look at panel B, by further examining the top 20 ranked genes from the original method, we can see that some genes like COL25A1 lose significance and are no longer considered significant, whilst other genes move up in rank as a consequence, like ChRNA1. Some genes shift in rank only slightly, such as COL9A3. If we look at panel C, this change in gene rank and in the number of significant genes influences the pathway level findings. Using the original method, we see that pathways such as mitotic cell cycle, collagen formation, nuclear division, NCAM signaling, ossification, protein catabolism, amongst many others are found. But when we account for directionality, some pathways are lost, such as collagen formation, mitotic cell cycle. Almost entire themes are lost, NCAM signaling, whilst some pathways are fully retained without a single, or some themes like protein catabolism are fully retained without a single pathway lost. And some pathways are newly identified that previously could have been missed. such as integrin cell surface interactions and positive regulation to protein ubiquitination. So this modified method provides more confidence in genes and pathways identified as impacted by HOXA10AS and can be used to facilitate biomarker discovery and biological interpretation. So to summarize, we provide a flexible framework to integrate data by additionally defining the direction of interaction of genes in different data sets. This tool is applied to a HOXA10AS case study to integrate differentially expressed genes and prioritize key biomarkers and pathways implicated in HOXA10AS function. The modified active pathways method can be applied to a variety of different cancer datasets and analyses where p-values and directionality information can be acquired. In the future, we will develop active pathways into an accessible and interactive web application to identify enriched pathways from multiple datasets. And with this, I'd like to acknowledge and thank the Riemann Lab, my committee members, as well as Medical Biophysics and OICR for funding. Thank you so much for listening to my presentation.